this video, we will discuss the retina, the low level of visual processing. The retina is the brain's window on the world. All visual experience is based on information processed by the retina's neurocircuit in the eyes. The retina is a thin sheet of the neurons composed of 5 major cell types: photoreceptors, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. These types are arranged in 3 cellular layers separated by 2 synaptic layers. The photoreceptor cells absorb light in the outmost layer and convert it into a neurosignal, an essential process known as phototransduction. These signals are passed synaptically to bipolar cells, which connect to retinal ganglion cells in the innermost layer. Retinal ganglion cells are the output neurons of the retina, and their axons form the optic nerve. In addition to this vertical pathway from sensory to output neurons, the retina circuits includes many lateral connections provided by horizontal cells in the outer synaptic layer and amacrine cells in the inner synaptic layer. In this retina session, we discuss the important aspects of retina function: sampling, phototransduction, 2 steps of pre-processing, and adaptation. Also, we will discuss both the neuromechanism by which they are achieved and their consequences for visual perceptions. The sharpness or quality of the retinal image is determined by several factors: diffraction at the pupils aperture, refractive errors in the cornea and lens, and scattering due to the material in the light path. The fovea is where vision is sharpest and corresponds to the center of gaze that we direct towards the objects of our attention. Because the density of photoreceptors, bipolar cells, and ganglion cells is highest at the fovea, the light must generally cross several layers of the cells before reaching the photoreceptors. But in the center of fovea, called foveola, the outer cellular layers are pushed aside to reduce additional blur from light scattering. The retina contains another special site, the optic disc. The optic disc is the region where the axons of retinal ganglion cells converge and extend through the retina to emerge from the back of the eyes as the optic nerve. By this structure consequences, this area lacks photoreceptors, which corresponds to a blind spot in the visual field of each eye. Because the disc lies to the nasal side of fovea to each eye, light coming from a single point never falls on both blind spots simultaneously, and thus, normally, we are unaware of them. We can experience the blind spot only by using one eye. Close your right eye and fixate it across with the left eye. Move your screen or head about 30 centimeters from your eye and move it slightly nearer or further until the circle on the left disappears. Now, use your finger and move it sideways from left to right over the circle. You can see the fingertip is disappeared when it enters to a circle. This tells that the blind spot demonstrates what blind people experience. It's not blankness, but simply nothing. But after crossing the circle, you can see the fingertip again. This explains why damage to the peripheral retina often goes unnoticed. First, there are 2 types of photoreceptors, rods and cones. All photoreceptor cells has a common structure with 4 functional regions: outer segment located at distal surface of the neuroretina, the inner segment located more proximally, the cell body, and the synaptic terminal. The rods and cones are distinguished by their morphology. A rod has a long cylindrical outer segment within which the stacks of discs are separate from the plasma membrane, whereas the cone often has the shorter, tapered outer segment, and the discs are continuous with the outer membrane. Rods and cones also differ in function, most importantly in their sensitivity to light. Rods can signal the absorption of a single photon and are responsible for vision under dim illumination, such as moonlight. But as the light level increases toward the dawn, the electrical response of the rods became saturated and these cells caused to respond to variation in intensity. Cones are most less sensitive to light. They do not contribute to night vision but also solely responsible for vision in daylight. Their response is considerably faster than that of rods. Primates have only one type of rod, but three kinds of cone photoreceptors, distinguished by the range of wavelengths to which they respond. The human retina contains approximately 100 million rods and 6 million cones, but the two cell types are differently distributed. The central fovea contains no rods but is densely packed with small cones. A few millimeters outside the fovea, rods greatly outnumber cones. All photoreceptors become larger and more widely spaced towards the periphery of the retina. To understand how the sampling of visual information on the retina works, we need to go a bit deep into the molecular level of photoreceptors. As in many other neurons, the membrane potential of the photoreceptors is regulated by a balance of membrane conductance to sodium and potassium ions. The transmembrane gradients of these ions are maintained by metabolically active pumps. In the dark, sodium ions flow into the photoreceptor through non-selective cation channels that are activated by the second messenger cGMP. Absorption of a photon by a pigment protein set in motion a biochemical cascade that ultimately lowers cGMP concentration, thus closing the cGMP-gated channels and moving the cell closer to the potassium equilibrium potential. In this way, light hyperpolarizes the photoreceptor. Most of these knowledge derive from studies of rods, but mechanism in cones is very similar. The photoreceptor rods has a visual pigment called rhodopsin, and rhodopsin has two components. The protein portion, opsin, is embedded in the disc membrane and does not absorb light by itself. Retinol is a small molecule and is covalently linked to a lysine residue of opsin. Absorption of the photon by the retinol causes it to flip from the 11 cis to all trans structure. This reaction is the only light-dependent step in vision. The change in the shape of the retinol molecule causes a conformational changes in the opsin to an activate state called metarhodopsin 2. The first step of phototransduction, metarhodopsin 2, leads to the closure of cGMP gate channels in the plasma membrane. This is the second step of phototransduction. This channel closure hyperpolarizes the membrane and reduces the rate of release of the neurotransmitter glutamate. cGMP is produced by glutamate cyclase and hydrolyzed by PDE enzyme. In the dark, the PDE activity is low, the cGMP concentration is high. and the cGMP-gated channels are open, allowing the influx of sodium and calcium ions. And the light rhodopsin is excited by absorption of photon, then activates transducin, which in turn activates the PDE. cGMP level drops. The membrane channel closes and fewer sodium and calcium enter the cell. The transduction enzymes are all located in the internal membrane disk, and the soluble ligand cGMP serves as a messenger to the plasma membrane. The photoreceptor's response to a single photon must be terminated so that the cell can respond to another photon. Metarhodopsin 2 is inactivated by an enzyme called rhodopsin kinase, which blocks the interaction with transducin. In addition, this mechanism is an important feedback mechanism that ensures that large responses are terminated more quickly. Thank you for watching. That's all for the first part of Retina series. The next video will be the second part of the Retina, start with pre-processing of the visual information. See you then.